We can separate the activity of GABAergic synapses from that of glutamatergic ones by applying a blocker of AMPA kainate and NMDA channels in the bath, such as APV and CNQX. When researchers record spontaneous GABAergic currents from a single neuron, they get information on the activity of GABAergic neurons afferent to the one being recorded. Students often think that only glutamatergic excitatory neurons are active, but inhibition is also present spontaneously.
I record GABA-A currents in whole cell configuration and in voltage clamp mode. The chloride ion's reversal potential is negative 60 millivolts. I hold the membrane potential at positive 10 millivolts. This holding potential has a double advantage. First of all, the GABA-A currents have a large amplitude. Additionally, the cation currents are not visible because they reverse at this holding potential. Under these conditions, GABA-A currents are outgoing, i.e., here there are upward deflections of the baseline because chloride ions enter the neuron. A time base of about 250 milliseconds provides an indication as to the long duration of the currents and their recurrence frequency. We see a recording of spontaneous GABA-A currents over time here. These currents have a variable amplitude on the order of tens of picoamps. This amplitude is variable because the number of GABA-A synapses active at the same time is not always exactly the same. The more active GABA-A synapses there are at the same time, the greater the amplitude of the current. We launch automatic current analysis in the software and then validate this analysis manually. The parameters of interest to us for each current are the amplitude, i.e. the magnitude of the deflection measured here between the yellow point and the red cross, the current rising duration, which we will measure between the yellow point and the red cross, and the current falling duration, which we measure between the red cross and the violet point. Finally, we will be able to measure the time that elapses between two consecutive GABA-A currents. In conclusion, spontaneous synaptic GABA-A currents recorded under physiological chloride ion concentrations provide insight into the GABAergic connection rates of the neurons being investigated. This enables us to understand the activity of the GABA-A synapses afferent to the neurons being recorded under optimal network activity conditions.